And so today I'd like to talk about our approaches to obtain high productivity and shortened timelines from development to commercialization at BASF. So just for those who are less familiar, BASF is a global chemical company headquartered in Germany with over 100,000 employees worldwide. As one of the largest chemical companies, our chemistries are used in almost all industry segments, including things from surface technology, nutrition and care, all the way to agricultural solutions and more. So part of our success is due to our investment in innovation. So we have approximately 10,000 employees in R&D alone, with an annual spend of about $2.3 billion just in research. So not just investing in innovation, but also investing in sustainability specifically. So BASF is looking to reduce both our resource and carbon footprint. And we have a two-pronged approach for this, both looking at carbon management to reduce our greenhouse gases, as well as investing in a circular economy. So looking for recycle or renewable-based feedstocks, as well as closing material loops. So within BASF Biotechnology helps contribute to sustainability by providing bio-based solutions to meet market needs. So this could be in the form of biologicals, small molecules, or protein products. And we do this through our end-to-end capability platform, where we have core competencies in everything from discovery and screening and engineering, strain development, bioprocess optimization, all the way up to scale up and launch. In addition to our core competencies, we also have invested heavily in automation and digitalization capabilities, which enable us to do high throughput screening and data analytics and up to tens of thousands of variants per campaign. If you look at BASF as a whole, we have over 25 years of biotech experience with over 200 employees in R&D, 50 of which are located right here in San Diego. So for the next slides, I'm going to highlight our core competencies, specifically focusing on our protein development pipeline. So first, we start with a customer need, and then we go to look to discovery. And so we have multiple approaches that we take to do discovery that we've been into two main categories, either a function-based approach or a sequence-based approach. So for the function-based approach, this is when we typically don't have a known protein class, which we think would fit for the customer need. And in this case, what we would do is screen our internal isolate or metagenomic library collections and look for activity on a surrogate assay. This could be either on an agar plate or in microtiter plates. Once we find a strain or a library collection that has the activity of interest, we then have multiple omics technologies to identify the protein or proteins of interest that are causing this activity. We will reclone them, go through primary screening and hit confirmation before doing further optimization. For the second bucket, for the sequence-based diversity, this is when we do have a protein class that we think may meet our customers' needs, and therefore we will use a sequence-based approach. We can search both public databases as well as our extensive internal sequence collection, both from the strain isolates as well as the metagenomic libraries. Again, once we identify the diversity that we want to screen, we reclone these, go through our primary screening campaigns, again, identifying our primary hits before further optimization. So go next slide. So this is just an example from a sequence-based approach where we're looking at an enzyme class. In here, in the green dots, if you can see them, are the known diversity within that enzyme class, highlighted with all these green dots. And what we would do is we take representative samples of these blue dots here that represent different sequence diversity in that space, and go through our primary screening. When we do our primary screening, what we'll do is look for multiple parameters, including expression, stability, and application performance. This gives us our final hits, and you can see for this example that our hit sequences all kind of cluster in one sequence space. So what we can do there is then go and relook at our sequence space, more focused on this cluster, and identify internal sequences that have more desired performance for all of our different parameters, now in that localized cluster. Go next. So once we feel like we've exhausted the natural sequences, we at BSF have an extensive history of being able to do genetic engineering to improve those proteins above the natural diversity. So this can be for many different characteristics, either for performance or manufacturing robustness. In the case of performance, we have experience doing things like temperature and pH optima, as well as substrate specificity. For manufacturing robustness, these are engineering enzymes, proteins to improve protein levels, recoverability, or extended shelf life. So once we have our final enzyme with the performance characteristics that we need in order to make a viable product, the next thing we need to do is go into host engineering. Go next. So here we have many different options available to us. We have a lot of expertise and experience in many different host systems, including gram-positive and negative bacteria, yeast and fungal strains. And when we choose our host, we take many things into consideration. So we can look at both the host as well as the protein of interest and the final application. So we need to know like what yield predictions that we expect, what protein quality we need, and importantly, what's the regulatory impact for the final application, and then look at the overall risk. And so once we take all these factors into consideration, we will choose a final host system, and there we can go through strain optimization. And this is where we work closely with the bioprocess team to make sure that we're engineering strains that will work with the final production process in mind. Okay, next slide. So zooming in and looking at Pichia in particular, what tools do we have available? So for Pichia, we have multiple methanol-free promoters, which I'll describe in the next few slides. We also have demonstrated signal and DNA design optimization. We have several host strains that we can use and further optimize using various techniques, including adaptive lab evolution, and we're continuing to develop these toolboxes now. So as I mentioned, we have described in the past our methanol-free system. This was a process-first approach, where we first developed fermentation processes that were easy and robust, lowered biomass, and used cheap media components. We then identified promoters that were highly expressed in these process conditions, either constitutive or upregulated, and then recloned those and validated that they could drive exogenous gene expression in our process. And here's just an example on the right-hand side of a SDS page gel of a single gene driven by either our internal promoters or the AOX methanol induction system. In the past, we've demonstrated this primarily with industrial enzymes such as lipases, amylases, and xylonases, but we have examples more recently on the next slide that we've demonstrated this with other challenging proteins. And so on the left-hand side is an example of our ability to express a single domain antibody. We've done this both in the microtiter plate as well as the bench scale fermenters. And here is just the unpurified supernames. You can see a nice clean single band. We can't disclose the final titers for this, but we can say that we're, with minimal optimization, that we're reaching single-digit gram per liter whole cell broth with this product. In addition, we've also got an example with peroxidase. Here, this is an early study just in the microtiter plates. And here you can see that even though we tested multiple methanol induction processes that were reported in the literature, that our methanol-free process was as good or better than the methanol induction system. So again, demonstrating that our methanol-free process seems to continue to be beneficial for multiple challenging proteins. Next. So after we have our strain and our protein, next and final we go into process optimization. And here our team has experience in everything from microtiter plate all the way up to pilot scale. And they've been able to demonstrate scalability and predictability at every level in between. In addition to just the bioprocess itself, they also have experience in downstream processing optimization, so to improve recovery, product recovery workflows. So here's just an example, again, of our methanol-free system. In the fermenters, on the left-hand side, you can see at the bench scale, a comparison expressing lipases, where we can see that the methanol-free process is as good or better than the methanol induction system. And then when we translate this to the pilot plant scale, that it scales quite nicely. And I should mention here that these titers are given as whole cell broth. If we look at the supernate, we have about twice as much. And then during this fermentation process, there are multiple drains that go on due to the feeding. And so if we looked at the productivity titer per working volume, it would be approximately 50% higher. Next. So even after this initial studies, we've been able to demonstrate that we can continue to improve volumetric productivity through process optimization. Again, here's an example from the lipase. And importantly, that we see that the titer increase is linear to about 140 hours and is continued to be maintained until 168 hours at least. We haven't gone beyond that yet, which suggests that this system not only can enable fed batch, but also potentially be developed for continuous fermentation for further productivity gains. So just as a quick summary, that we have our methanol-free system at BASF that's been patented, and it looks to be as good or better than the methanol induction for many challenging proteins. We've also demonstrated that it's scalable and has the potential to be developed for continuous fermentation. And we're continuing to develop more strains and molecular tools to further improve upon this system to improve titers even further. And then, as before, licensing options are still available. So with that, I'd just like to thank our entire team at BASF. We really have a multidisciplinary approach that allows our success, allowing us to think the whole process in mind, and really a lot of team members involved in all of this. And just quickly, before I end, one more slide. We'd like to announce, as of a couple of weeks ago, that we've officially launched BASF Bioservices. And this is where we provide services throughout the process, from discovery and screening all the way to bioprocess optimization for third parties. And if you want to learn more, you can talk to myself or Shichu, or you can reach out to us at bioservices@basf.com. And that's all I have.
Protein production in Pichia versus other yeasts. How good is it? Is it better than other yeasts? And with so much advances in the technology now, with new promoters, new strains, etc., I'd like to see a comparison.
I can say that we haven't ever done direct comparisons. You can see the titers, and we can also say that it's very protein specific, so you really want to do head-to-head comparisons for your protein of interest if you really want to see how they compare.
Thank you for the nice talk, Melissa. kind of taking a step from Surash's, when you've looked at the same protein in different hosts, do you have any kind of guidelines or thoughts about which proteins do well in Pichia as compared to the other or do better in Pichia compared to other platforms?
Oftentimes, things that we look at are where the protein comes from. So a lot of eukaryotic things do better in eukaryotes, things like that. And it really depends also on what kind of titers do you need. So for Pichia, as you mentioned, another yeast, we do have kind of a ceiling sometimes compared to other bacterial or fungal strains. And so it really depends on a lot of factors, the purity that you need, the market that you're going to. So it's not necessarily just a pure titer calculation.